Cool. Well, welcome everybody. Thanks for joining us for our 11th iteration now of the CMFI or MassSpec seminar series. And yeah, it's a great pleasure to have Ming Wang today give the seminar. So for those of you who do not know Ming, Ming is basically the brain behind GNPS, who designed and maintains most of the software under the hood there. So Ming did his undergrad in computer science at UC San Diego before he then did a PhD in the group of Nuno Bandeira on computational mass spectrometry, mainly in proteomics at that time, but then somehow I think he got fascinated by small molecules and particular computational metabolomics, which then also led him to do his postdoc in the lab of Peter Dorstein, where I guess I was fortunate to overlap for a little bit with Ming. And yeah, now I think Ming is on the jump to start his own lab very soon. So very excited and looking forward what will come out of your lab there. And yeah, for now, I'm also very excited to hear about the GNPS dashboard and in particular new developments on repository scale mass spec searches. So yeah, without further ado, Ming, thanks again for doing this today, and yeah, looking forward to your talk.
Yeah, thank you, Danny. And yeah, so yeah, Danny and I have worked together for a long time. So and I don't think that's going to change. I'm moving just a few miles, down the road in the coming weeks. So it'll be exciting. It'll be exciting. But thank you all for kind of showing up. And so what I wanted to talk about today is it'll be a hands-on session. And we'll go over how to visualize and explore some mass spec data in the browser. So we'll go through some of that. And we'll use this as a springboard to do some repository scale analysis and a few new tools that I've been developing. A little bit of hands-on. We're still writing up the paper, but I think it could be useful. So I'm curious to see everybody's feedback for everything that we talk about today. So anyway, thank you again, Danny, for inviting me. And I'll show you what our outline is. So first, we're going to get started with a GNPS dashboard. Then I'll do a little bit of explanation, but it's more a little bit of voice exposition, and then more hands-on. So feel free during all of this to ask questions in the chat. If you're behind a little bit, I think, Danny, did you send everybody the presentation file?
No, but I can provide the URLs in the chat.
Okay, yeah, let's do that. Let's do that. It's good to do the catch-up links, and then we'll keep going. But okay, so the first thing I'll talk about is GNPS dashboard, and so I'll start off with a frustration that I had for myself. And so I'm a computer scientist, and not particularly a mass spectrometrist, but I sit close to a mass spectrometer, so I talk to people who tell me things. But the number one trait, as most computer scientists will tell you, is we're very lazy. I really don't like doing anything. And so one of the problems was whenever we wanted to look at mass spec data to figure out problems with algorithms, we're just going to get some intuition about what's going on. I really hated downloading the files, number one. I hated converting the files. I hated installing software. Just to, especially, I mean, there's a need sometimes for MZMine and tools like that if you're doing large scale data analysis or feature finding and things like that. But all I was interested in was looking at a particular peak for about 5 seconds and spending a couple minutes to do something, to prep to look at something for 5 seconds was really, I really did not like that. And I was talking with Danny about this, and this must have been 18 months ago when we started this project, which is pretty new as far as research projects go. But he also expressed kind of a similar frustration but he's not quite as lazy as I am. And so we were like, what if it feels like GNPS is online? It feels like this is the natural place to just be able to do this seamlessly with a single click and never have to think about that process again. So that's kind of where we came from. And so what we came up with is this GNPS dashboard that kind of sits at the nexus of a lot of different tools and resources. And so first, if you're doing some data analysis of GNPS, you can go directly into the dashboard with your raw data, no matter what kind of analysis context you're doing. If you also have seen public data at Metabolized GNPS or Metabolomics Workbench, you can visualize that directly. So any new publications that come out, or you have your own data, you want to drag and drop it without installing anything, you can go ahead and do that. And similarly, imagine that you do some analysis and you want to share with somebody else. I mean, I don't know how many times you all have sent a screenshot to somebody or told somebody, click here, oh, open it this way, click here, which is not a particularly fun endeavor, especially in the times of remote work. And so the ability to share your analysis and your visualization with somebody else and with the wider community in a publication or on Twitter and social media, that's something that we do all the time. I send stuff back and forth with Danny. Say, look at this cool feature, and he sends something to me, bragging about how cool his chromatography is and how sharp the peaks are. So it's partially a vanity thing between us, but as well as for us, we integrate downstream analysis tools directly into the dashboard, so it's easy to do additional things and expand the capabilities. So before we get too into the weeds, that's kind of a high level thing. I'll walk you through the user interface. It's relatively straightforward, but for the very first time ensconcing, Danny, if you could grab the link right here and put it into the chat.
Yeah, I already did.
Okay,
so
awesome.
You should just see the URL there.
Yeah, so go ahead and click on that. That brings you to a particular analysis that I've done. And just to walk you through the different parts of it, there's portions where you can select what data that you want to look at. In the top left, identify by one. On the right top, you have more options to define like extracted ion chromatogram, how to do the formatting, and visualize MS2, rendering options. Number 3 is this 2D LCMS heat map. So the x-axis is retention time, on the y-axis is M/Z. And then you can see there's little blue markers that represent MSMS acquisition points, as well as if you remove those, you can remove those blue markers and you can see a heat map of the intensity across your chromatogram, splayed across M/Z as well. On 5, you actually have your extracted ion chromatogram plots as well as your MS2 plots right below that. And under 4, you have your TIC, and then that can interact with your MS2 plotting there. And so we can walk through this a little interactively. So let me pull that over here. And so we can see again, here's the file selectors on the top left, the XIC options here, and they're already pre-filled for you. And so if we put in several files, or one file for you, and we put in the XIC for a particular molecule that I'll go over in a second, with the M/Z and the retention times already set. And so what you can see down here is that it's zoomed in on a particular peak, as well as the MSMS that's shown up here. And you have the TIC down here. So it's a pretty complex thing going on. And there's a lot of interdependence. If you click on a particular point here, on the left-hand side, it'll update the XIC and the MSMS. So it's kind of everything kind of links together. If you click down here on TIC, it'll show you an MS1 peak in this MS visualization. So we'll walk you through a little bit more of the features of this. And so I'll play around with a little bit in just a second. But one of the cool concepts that we think is pretty unique, and things that you can't really do in other software, specifically because the GNPS dashboard is this online web resource, is sharing and synchronization. And so there's kind of 3 levels of synchronization that are possible. And the first one is just simple URL sharing. So I can copy a link out, and then I can send to somebody else, and they can see exactly what I'm seeing on my screen. And so there's no more futzing with, oh, please click here or zoom in on this. they can see exactly what you see and build upon that visualization. And so that's one way to kind of asynchronously share what you're looking at. The second one, and we're moving into this synchronous visualization, is called this leader follower. And so we're kind of borrowing some of these ideas from the Google Docs and these online document editors, where if I change something in my visualization, all of my followers can see exactly what I've changed in real time. And so this is, for in our view, pretty useful in training. So if you have new people in the lab and you want to guide them through their first analysis in mass spec data, you can be the leader and you can have them follow along. Similarly for classrooms. And so the nice thing about this is it's kind of a supercharged, screen sharing session where they follow along in real time and they can stop following you and build upon that visualization as well. And kind of the third one, this full synchronization mode, this is really kind of the Google Docs online document editor. All the people in the session will see exactly the same thing and everybody can edit what's going on. And so it's kind of bi-directional across everybody in the session. And so that's kind of interesting and fun, but we've done more of the first two, definitely for classrooms and for publications, and just discussing it between collaborators, especially like sending links on Zoom or Slack. It's kind of fun. Danny and I just send things back and forth all the time, and we change a few things. But anyway, that's kind of a few other features. And just for clarity, if you do want to share a link to somebody, there's a little button in the top left, right under the file selectors, that says copy URL to this visualization. Click that, it'll give you a big URL. You can send it to a friend, put it in a manuscript, as well as bottom to the right of that, in case you can't put it in. There's a question about expiring links. So on this left-hand link, this won't expire. So this will be valid. It encodes literally everything about your visualization in the URL. That's why it's kind of a mile long. So the problem is you can't actually put that in something like the Zoom chat. It doesn't like that. Manuscripts are not a particular problem, but Zoom chat is. So that's why we have this short temporary URL. We basically build our own temporary tiny URL, and you can take that. It'll be valid for an hour, and it actually expands the full URL, and so you can use that to put it into something like Zoom or like GChat if you're using that, if they don't like these monster links. And so the real question is about, does your data expire? And so it depends on where your data is coming from. And so if you upload it manually yourself, so you can drag and drop, that will expire after a few weeks. And so don't put those links in a manuscript. But if you deposit your data at a data like a metabolomics data repository like Metabolites or Metabolomics Workbench or GNPS Massive, those will stay around as long as the resource stays around. So that's kind of not your problem in a sense. So those are more effectively are safer to put in publication. Any other questions so far? Okay, cool. So what we're going to start looking at now, kind of a hands-on portion, is there is a data set pulled out from this particular article that we published a couple years ago that we're looking at bile acids, some new conjugated bile acids that affect that are produced by the microbiome that modulate kind of host activity. And so one particular molecule is this bile acid that's conjugated with phenylalanine. And so this is a newly discovered bile acid in this publication. And we're going to kind of look at it and use this as the backdrop in the dashboard, as well as a few other things that we want to start looking at. And so again, if you click that example link, it'll be focused in to exactly that particular molecule to look at the extracted ion chromatograms and the MSMS. So we can see on the extracted ion chromatograms in your visualization that there's kind of a peak here. It's not super abundant, but it is there. And we can see these little red X's in the XIC, and these indicate MS2 acquisition events. And so you can kind of see that it was acquired twice. You can click on these to see, if you click on the red X's, it'll show you below with the specific MSMS that you clicked on. And below you can see the MSMS. And since we have the MSMS, let's assume that we didn't actually know what it was. And we can actually do some cooler things in the GNPS dashboard directly. And so we can start answering these questions. Now, first, can we identify this compound in the spectral library? Do we know what actual molecule this is, if it's been known before? And we can answer that question. And then number two, kind of a more interesting question is, where else have we found this molecule before? Even if we didn't know what the compound name and structure were, can we ask kind of more contextual information and leverage all this public data? Have we observed this same MSMS molecule or MSMS spectrum in other data sets that give us an idea of how broadly and what kind of data sets and samples do we actually find this particular molecule? So we can start answering both of these questions. And so the very first thing, let's see a question. Is there a way to see sample information about samples? So if we're looking at public data, there is. And so that's a tool called Redo. And so we'll take a detour a little bit into that in just a second. And so what we'll first start doing, we'll move on to the second section of searching public data. But whenever you visualize an MSMS in the GNPS dashboard, there's a couple of links that start changing below it. And so one of the first one is called right here is GNPS library. And for every single MSMS that you visualize, we actually scurry out and we do this all in real time. We kind of call this the prefetching mode. But we actually search that MSMS against all public GNPS libraries before you even ask for it. And so what you can see in the parentheses here is it tells you the number of putative hits that it actually gets against the spectral libraries. And so if we hop over to the dashboard here, we can kind of, I'm visualizing the MSMS right here. We can see that it says 23 putative hits. Now, if you click on this, it brings up a little pop-up screen. It actually embeds another website in here. It's called we call it fast search. So not a particularly creative name. But what it will do is it searches that MSMS for you against all the spectral libraries by default. And it provides you the table of all the matches already ready for you to go. And so, let's go back to the presentation. You'll see this list. And so you can go through this list and you can actually scroll to the right. It's kind of wider than my screen is. And you can see the putative hits that it could be. And so if you all take a quick look at that, you'll be able to tell what kind of molecule this might be. And so this is not particularly interesting because we already know that it's phenylalanine conjugated bile acid, and that's what kind of shows up at the very top. But this allows us to do that. You can click on any MSMS spectrum within your entire file and immediately have a putative library hit. And so if you want to do a little bit more due diligence, you can select a row, compare your spectrum to the library spectrum. So the rendering takes a little bit. But you can kind of see how well it matches up. We can look at the raw data as well. But anyway, that's kind of one thing that you can start looking at. Any questions so far? Cool. And so that allows us to do library matches to known things in the library. But the libraries are pretty small, right? And we can't identify everything. So how do we start taking advantage of all this public data? And so just giving you an idea before we kind of go through the mechanics of it. So this is a tool called MASST. And so this is actually a newer unpublished version of MASST. And so I don't know, Peter might have actually talked about this when he was presenting, but the idea is we have like 40 terabytes of public mass spec metabolomics data. And so this is comprising over 1 billion different molecules in like 300,000 different samples. And so it's a ton of data. And so in the same way, if you do a lot of genomics analysis, you can take a DNA sequence and ask what organism, what species, what sample types have I ever found have this DNA sequence with a tool called BLAST. We can do a similar approach here with an MSMS spectrum. And then that gives us a lot of contextual information against all this data. Again, what instrument, what sample, what organism, what kind of metadata is present. And so we can kind of do that very quickly, whereas before we were doing it about 15, 20 minutes. Now we can do it in about 15 and 20 seconds. So what you'll be able to do in this interface for the library search interface is you'll just change the library name. Before it was GNPS library, but now we can do the drop down and select GNPS data index and click the search button. So I'll walk you through how to do that right here. So if we go back to the top and we change the library name to GNPS data index, and we click the search button, it'll do its little thing down here. And it'll tell us, okay, we found this guy. Actually, it doesn't give us a summary. There's about 10 per row on 173 pages. So we found this MSMS spectrum in about 17, we found the same MSMS spectrum 1,700 times across many different data sets. So you can see like the data set identifier here and then the match scores and all that stuff. We found it in the same data set as at the very top, basically the same file, so not super surprising. But if we scroll a little bit down, we can start getting a little bit more summarization data. So there's several of these tabs. And so we won't look at this first one, but we look at the second one. And then this groups it by data set. And so we can see that it's found in about 60 different data sets. And we also summarize the data set accession, the frequency that it's found in, and the data set title here. And so it's a little bit truncated. So we have to, there's a few improvements to the user interface we got to make. But you can kind of start seeing that this particular metabolite, because we published, we thought it was made by the microbiome in the gut. We can actually see that it's also being found in the gut of some cheetah samples, some mouse fecal metabolites, some bile acid extracts, other mouse data sets, some stool from the IBD biobank. So we can kind of get it's very clear from this data set list that it's from all these kinds of microbiome gut samples. And so there was a question from Frank a little bit earlier about metadata. And so what we do have is you can I believe you can download, so I don't know if you can download these tables. We got to make a button to download these tables. But we're working on making sure that at the file level metadata is included. And so where we would include this is a resource called Redo. And so this has sample information from 50,000 samples. This is something the community has curated. And so what is the official number now? So 53,000 samples have consistent control vocabulary metadata. So for example, if we look at health status is one. So things like chronic disease, are they healthy or not unhealthy? We can look at body parts, where it came from. And so there's files here. And the nice thing about this is you can download the whole database as a big Excel spreadsheet if you want to kind of look at that and play around with that data yourself. But any questions so far? But cool. So, and if you wanted to use this, you can put in any of your data. You can actually enter your own peaks here if your data is not in the GNPS dashboard or publicly available. So QA, QC for data screening. So that's a good question, Frank. So it depends on what you mean by QA and QC for screening. So one of the things that you can do, and we do this quite regularly, is let me pull up another LCMS file here. So this happens to be a QC run that Danny has run that he puts in his lab, that he runs a 6 mix of a bunch of standards. And so what you can do that makes it nice and easy to do is if you put like controls in all your samples, or if you just have QC runs with a QC mix, what you can do is you can set presets for your extracted ion chromatogram, and you can inspect what's going on really, really quickly across a bunch of your samples. This happens to just be one sample, but I think what we can do is, if you have multiple samples, like this happens to be QC0 and QC1, you can put multiple in here and just kind of scan the extracted ion chromatogram across all of them in a really, really fast way. And if this is your cup of tea, there's heat maps that allows you to even summarize it. So this happens to be a 271 MRZ, and then you can have like 50 files here, and you can just glance at the heat map, and you can quickly see which one is deficient in a particular QC. So that's one way that you can at least QC what's coming off your mass spec. And then the nice thing, especially if you're running a bigger lab, one of the things that I think is pretty interesting is if you are the supervisor and somebody's running something, they can send you a daily report as a link to this visualization. And then you can look at all the QCs run the previous day and see, is that within bounds? It's not just saying yes or no from kind of the technicians, but rather it's the evidence and the data backing that assertion. So, it's something you can put into your electronic lab notebooks, and you have a data record of everything. So you never have to just say, well, they said yes, but now you have the data behind it. So does that make sense, Frank? Okay, so that's one thing we can do for analyzing public data. So yeah, this is walking through kind of all this. So you can group by data set here. But the name of the game for us here is that it's really, really fast. So you could ask many different questions about many different molecules in rapid succession. Whereas before, if you had to wait 20 minutes to get to these kinds of answers is a little bit harder. And so one other cool feature that I want to kind of show off to you guys, especially because you all have had experience with the GNPS or with GNPS in general, and you know about molecular networking and things like that. So let's go do the search here. And so initially, when we did the MASST data, the searching across all the public GNPS data with MASST, it was looking for exact matches. Where have we seen this same molecule before? And so one of the kind of hidden modes here that is kind of fun to play with is you can do an analog search. So you can find related compounds to your molecule, but at a repository scale. And so you can think back on molecular networks where you try to group molecules together based upon similar fragmentation. It's borrowing that same idea, but we're extending this to an entire repository of billions of molecules. So it's like, are there related molecules, not just in my data set, but across everybody's data sets? And so I think it's kind of cool, especially if you're looking for like metabolism products or shunt products for biosynthesis and things like that, or chemical degradation. So we turn that on. And here these are the bounds for mass delta. And we click search. And this should also be relatively quick, but just not quite as quick as the exact mass searches. So I actually haven't run this, so just popped into my mind. So let's fingers crossed. So we'll wait a little bit while this churns a little bit. And I'm actually kind of curious. It's all running on a humble little box. So if you all want to try it too, be my guest. It'll stress our system. So I'm always looking to see how well everything works. And so we're done. So here, before we had 1,700 results for exact matches. Now we have about 3,000. And so what we can start seeing here, if we scroll down a little bit, before this first chart was super uninteresting because what this first chart tells us is the mass delta between all the hits that we found and our molecule. And so it was uninteresting because we were looking for the exact same thing. If we're looking for this exact same thing, the mass delta is going to be 0, kind of by definition. But now we're turning on analog search mode. And so kind of where this plot really came about and kind of how I thought about it is when we look for post-translational modifications of bottom-up proteomics. But we can take the same plot and reuse that for structural modifications for small molecules. And we can look at the histogram here and how we see a lot of molecules that are around 17 to 19 Daltons. So that's probably adding, I guess, water, something like that. Some things that are around 36 I guess 2 waters, I have no idea. And somewhere around 45 to 46. And we have a little chart here to look at the relative frequencies. But this allows you one way, given spectral similarity, to start exploring very, very quickly how you can, what is the chemical diversity of this particular molecule across all public data. So anyway, I think it's kind of cool. Play around with it, something to think about. And it's another thing that stresses our servers. So that justifies we can get more of them eventually. So that's MASST and the GNPS dashboard. Did you guys have any other questions so far? Cool. So one of the other thing I'll talk about at the very end is another thing that I'm personally excited about. So we're writing up the paper now, but this is something I created over like kind of the last year, again, out of sheer laziness. And so one of the things, a lot of you are analytical chemists and mass spectrometrists. And so one of the things that you have in your heads, or let me take a step back, is mass spectrometry is a tool that allows us to measure a lot of molecules, right? And one thing that we know innately is that a molecule, whenever we measure on the mass spectrometer, it creates some sort of signature there, right? It has to. It creates some data that's reproducible and to some extent unique, right? So if there's peaks in the MS1 spectrum, there's fragmentation in MS2 spectrum if you've also collected something in MS3. There's also retention time information, ion mobility information. And so depending on the composition, it can change all of these different things. And we know intrinsically as a chemist what those patterns are. And so one of the kind of the things that Danny might come to me and ask is, hey, we have all this public data, or even within his own data set. Can we find, for example, all the carnitines in that particular data set, or across all this billion data set? And now when he says carnitines, it evokes, and connecting that to mass spectrometry, it evokes some knowledge that we all have, saying, oh, we know carnitines will fragment in a similar way because of the nitrogen that has intrinsic charge. It might have an 85 peak and a 59 MOZ peak as well. And so there's kind of this knowledge that we have in our brains that understands what kind of pattern might be present. However, just because we know that doesn't make it really, that's good. That's really good for us to know, but it makes it really, really difficult if we wanted to look for these kinds of patterns exhaustively. And so there's, you basically have to write your own code to do this. Or if you go if you change the kind of molecule, the class that you're looking for example, brominated compounds, it's a totally different pattern or a totally different part of the data structure for the mass spectrometry data. So you have to write some new code to do that. And then I don't like to do that. I don't want to, kind of repeat effort for every person that has the kind of slightly different question. And everybody's question based on what they're analyzing is a little bit different. And so there's a lot of customization, and changing code is just really not the way to go. And so I pose that kind of question to myself is there a universal computational solution that can be general for everybody, but also really powerful and easy, right? So easy is the name of the game. And so I think the answer is yes. So kind of the thing that I invented is called the mass spectrometry query language. And so this is a very computer science way of tackling this problem. I think a lot of mass spectrometrists, people in the field would create a nice user interface with nice drop-down menus and things like that. But instead, I go the route of well, what if we can just say a sentence and it does everything we want? And so basically, the key pillars of what we wanted to do for MassQL was it needs to be understandable. So you as a chemist need to be able to read and write in this language. So it's like pseudo-English, right? Or kind of pseudo-human language. It needs to be flexible. So there's a lot of different patterns and things that you all want to look for. And it needs to be expressive and flexible enough to capture a huge swath of what you're actually trying to find without having to jump through a lot of hoops. It needs to be scalable. So this is kind of the biggest thing. So maybe it wouldn't be the biggest deal in the world for you all to write your own R or Python code to do something like this for a particular pattern. But then the real trick at that point is maybe it'll work for one file, but will it work for 200,000 of them over a billion spectrum? Generally, probably not. It's just not your kind of the forte of chemists to build scalable parallel systems. And number 4, it has to be reusable. And so kind of this is a bigger point about if you write your own code, well, if you hand it to somebody else, they're not going to understand it. And for them to reuse it, it doesn't, you want to be able to share take that knowledge in your brain, put it down on paper, and then give it to somebody else and then you can, especially if you have a bigger lab, you can give it to a colleague, you give it to a trainee, and they can start doing real work and exploring data without having to teach them this full intricacies right off the bat about mass spectrometry, fragmentation, isotopic patterns, and things like that. And so again, these are kind of the design philosophy of the mass spectrometry query language. And so just walking you through a few examples. So Danny was interested in this family of molecules called aldehydes, and these are NRPS, PKS natural products. And so what he knew based upon the fragmentation and some examples of the data is that one kind of portion of the aldehyde family always created these fragments at 468 MZ and 660 mOZ. So he knew that would always occur. And so he was able to write a query that's kind of pretty simple along these lines. It says, let's look for MS2 data where there's an MS2 product peak at 468 and 660. So you can kind of add these conjunctions so you can have multiple conditions that must be simultaneously satisfied to be really specific. And you can add as many conjunctions as possible. And so that's one example where we're looking at the MS2 data. And so other patterns that you can start expressing in this language are patterns, and this is within the MS2, and so we can do a similar thing in MS1. So you can start saying, hey, I want to look for a particular MS2 peak. So you can express that. You can look for specific neutral losses from precursor within the MSMS. You can start looking for mass delta. So I guess depending on the terminology, it's called neutral losses as well. But gaps between peaks in the MSMS or the MS1, as well as start specifying the relative intensity of a subset of the fragments within an MS spectrum. And so this is particularly useful in this bottom case if you care about like distinctive isotopic patterns like bromination or chlorination, and kind of other people are exploring it with metal adducts and things like that. And so one example, a simple one that I'll have you guys do a hands-on about is looking for glycan-containing molecules. And so one thing that you generally find in glycan-containing molecules is that the glycan might fall off first. And when that falls off, you generally see a 162 mOZ from precursor. And so what I've written here is a very simple query where it's like, find me MS2 data where there's an MS2 neutral loss of 162.05. It's kind of pretty tight here. And so what we've built out for the MassQL is a special interface that we call it a sandbox. Basically, we're enabling you to query fixed data that gives you a little bit, kind of confidence in playing around with data, especially it allows you to search against GNPS spectral libraries. And so we happen to know the compounds, and we know the compound names and the structures. And so if you try to play around with a particular query, you can kind of see, does it manifest the kinds of molecules that you're expecting, or do you need to be a little bit more specific about it? And so what we'll do, let me put it in the chat here. So msql.ucsd.eu. So this is the website that you all can go to, and I'll walk you through a little bit of this. But let me also copy the query specifically so you guys can play with it. So let me put that here, and I will go there as well and show you the few steps that you need to do to get started. And so the very first one is for the file name, you'll need to go to gnps-library.json. So if you go there, that selects the specific library that we're working with. And then we'll also just paste in the query down here. And then we will also hide this very top window at the very top here. So that's for other visualization that we were looking at. And so what you'll now see is a little tab here on the right-hand side called, let me zoom out, called data exploration. And so this actually will show you all the subset of entries in the library that meet this particular criterion. So it's a neutral loss from precursor of 162. And so once you do that, what you can see here, there's a column called compound name. And some of these molecules you all can recognize, but or you might not recognize it. But if you can also go to the following pages. But is there any sort of pattern based on the compound names that you can kind of see that kind of stands out to you given what we were looking for in the mass spec query? Any takers? Any takers? So maybe let me zoom in on my screen in case it's easier to see. So what is the structural feature of a lot of these compounds that we're finding? It should be obvious, I guess, just from the previous slides, but just kind of from the evidence of the data. And you can Google the structures if you want to, if that helps you as well. I'll have Danny, I'm going to make you look at the pattern. Do you see anything here? So some of them are flavonoids, but a lot of them are from plants. And so with a lot of plant molecules, what do you usually see attached to a lot of these plant molecules? Again, it'll feel obvious and it'll feel ridiculous for me to ask, but I'm just wanting you guys to take that leap right there. Right, there's a glycan on a lot of these molecules. Again, not particularly surprising because what we were looking for and how we designed the query was to look for this 162 Dalton neutral loss, right? And that is pretty indicative of having a glycan in the original structure. But the key point here is that we took that query that we hypothesized should work and we applied it to real data. And the real data confirms to us that what we're looking for is indeed what we find. And so that kind of gives us some confidence if we applied this to our own unknown data, that it would pull out and throw out everything else that wasn't a glycan and enrich only for molecules that have a glycan on it. So then you can start that prioritize what you're looking for. And so kind of the goal here, or one of the applications that we uniquely are enabling is, imagine, before we were taking all this public raw mass spec data and we were asking, hey, have we seen this molecule in there before? Well, that's searching for that particular molecule, but we can broaden the scope a little bit, right? What if we wanted to find all glycan containing molecules across all public mass spec data and then visualize the chemical diversity in the molecular network? You can do that today. You can pick any sort of subset of, even if we can't identify any chemical subset that you can express that pattern that should be presented, pull that out and look and take a microscope from that bigger data and look at that full chemical diversity. And so that wasn't possible before, but now these tools give you that kind of capability. There's a couple of workflows that are behind the scenes that you'll actually have to run. But anyway, that's kind of the retailing of why this is kind of cool. And similarly for your own data, if you have a hypothesis untargeted, you might have tens of thousands of compounds, which isn't billions, but going through manually tens of thousands of compounds to look for a particular pattern is I don't like it. I'm not a big fan of that. And what was interesting Two is, as we're writing the paper, one of the new postdocs at UCSD, she cares about environmental work, and she did some untargeted mass spec looking for this flame retardant molecule. And so she manually looked through all this kind of untargeted LCMS data, and she published her paper with a couple new flame retardants. And then she made the data public. And then once she joined the lab, I was like, hey, you should try this MassQL thing. It's kind of new. And she was able to write a query, search her data, visualize, and look at only that particular subset of data, that compound type, and discover new flame retardants in her own data that she had already published on and had thought and looked exhaustively, but it turns out manually doing it was not exhaustive enough. And so that's kind of an argument that automating these things, codifying these things, really makes it, makes it a lot more thorough and finds what you were actually looking for instead of kind of giving up a little bit earlier, just kind of fatigue. And so I think it's kind of cool. If you want to use it, definitely let us know. And you have a question, can you do, okay, yeah, you can search your own data. A lot of people search their own data. So you just, if you create molecular networks, you can do it on your own private data. So upload whatever you want, and you can enter these queries. And you would have to run a workflow GNPS. So I'll send around kind of documentation, but you can search one file, you can search 100 files at once, and you can kind of go from there. And kind of going forward, this language, we're making it more universal, and universal in the sense that we're working with people at MZMind, we're working with people at MS-DIAL, and like OpenMS, and though they all have support now for MassQL. So you can write your query at GNPS right now, try it on your data, at GNPS. But what you can do in the future is you take that same query, like exactly the same query, and you can plop it over to MZMind or OpenMS, whichever piece of software you're analyzing your data at the moment, and execute and do the same operation. So then that means, if you have a friend that likes OpenMS and they develop a query there, they can give it to you and you can run it in GNPS. So it's becoming this international or like interchange of information that allows you to communicate a mass spectrometry pattern. And so one of the ways that we're encouraging this even further, especially if you're trying, for the first time, you might not understand what this means. We have a translator. So it takes a query that you might write and it translates it to a more human readable language. So here we write a translation in English, so you can give it to somebody new. And especially if you have other people in other languages that are not so fluent in English, some of the translations might not be so great, but if you want to contribute to making sure that it translates well, we welcome your feedback. But it just makes kind of the MassQL a little bit more accessible to other communities across the world. So this is kind of a thing that we're encouraging. And then this kind of helps you explain this, especially if you're a chemist, explain a little bit what's going on to maybe somebody that's not so deep. They might be chemists, but not so deep within mass spectrometry. So that's kind of the ecosystem of MassQL. But I think it's kind of cool, but we're happy to share like how other people have used it. As we're writing the paper, we collected about 50 use cases from people across tons of different domains to show kind of wide applicability. So happy to share kind of that SI, from natural products to metabolism, to metabolomics more properly, kind of human metabolism to environmental sciences, all across the gamut of applications. So, Frank, you have a question. Can this protocol be used to predict certain property of compounds? So that's on, I have no idea, right? So whenever you're looking for a particular pattern, that's what you're looking for. If that pattern means something from a toxicity or reactivity perspective, then yes. A lot of times just chemical structure might not be, you might not be able to tell, and you have to have some other functional assay. And so that's, talk to Danny about that. He loves talking about those things. But if there is some pattern in the mass spec data that tells you that it might be in a particular functional state, then yes. For example, like with Allegra, so Allegra Aaron at UCSD, and so she'll be at Denver or University of Denver soon. She cares about metal binding. So is a particular, for example, is a particular molecule stereophore. And so one of the things that we're looking for, especially across repository, is if we can find a molecule signature that is present and that it's bound to iron. So you see both of those patterns in the MS1, that tells us something about the functional characteristics of that molecule. And so that's a particularly special case because, you know, there's a pattern that shows it's actually doing something and bound. But in general, you might not be so lucky. But one case that you could start thinking about is, from an environmental persistence perspective, is if you look for, you know, this particular example I was talking about from Nina, looking for these flame retardants, it's called organophosphate esters. This particular class of compounds, we're able to look for that across all the public data. And so that number one, our first goal was to understand the chemical diversity and discover new ones. But at the same time, one of the things you can do is, because we search across all this public data, we know exactly which files and samples it was found in. And then we can connect that back to the redo metadata for what kinds of data, where was it found, what's the latitude, longitudes in rivers and things like that. So then it can tell you something about the geographical extent of the pollution, right? So that's another way you can kind of get at that. Or in the sense of the bile acids, we were looking at what kinds of data sets it was present in, and it told us, okay, it was found in mice, it was also found in humans. That tells us something about the translational component of that particular analyte or that particular class of compounds. And so you can measure these things kind of, there's so many, we've built out so many different tools. So it's like one tool doesn't answer all these things, but in conjunction, you kind of get these indirect measures and get to where you're going to. Does that sort of make sense?
Yeah, if I may, I guess I'm just interested because you mentioned that you found a new flame retardant based on this search. I'm just curious, like when you look for the new flame retardant, what kind of patterns or the like features you are looking for?
Right, it's flame retardants of the same compound class, and so I can't actually describe chemically what that class is, but it's variants of that same kind of core, and they just kind of they can change a little bit. So it's not like a totally novel flame retardant, but it might get chemically transformed, or there's a lot of different structural analogs in the manufacturing process, or it's being metabolized kind of in the environment, and we're picking those up in real data. And so it's rather than a totally new flame retardant per se, it's just exploring the chemical diversity of that particular flame retardant more exhaustively in a data-driven approach.
All right. Well, thanks so much, Ming. And before
there's one more question.
Yeah, we'll give it a second.
Okay.
We can like move on to like the more informal question and answer session soon. But yeah, like before we do that, I just want to like thank everybody for joining, and especially you, Ming, for giving this awesome presentation with like the super cool tools. And yeah, just a primer for next week, or like for 2 weeks, we're gonna have Hannes Link actually showcasing their flow injection analysis pipeline. So we've talked a lot about LC MS-based metabolomics, but he will actually showcase how we can get metabolomics profiles without doing any LC. So yeah, I'm very much looking forward to that, and also to the discussion now in a second with you, Ming. And yeah, everybody who's taking off, thanks again for joining, and hope to see you next time.